Welcome again, everyone, to our first GBM of the semester. We'll go ahead and get started with a few brief announcements before we invite our guest speaker to speak for today. So some brief announcements. This Friday, we do have an event that we are co-hosting along with a few other organizations on campus, as well as the Office of Diversity and Inclusion. So this event will be an open discussion with a licensed psychologist from the UIC Counseling Center. And the idea is to kind of discuss some of the recent events that have been going on in our country today and kind of talking about it and practicing some self-care with that. So very important event. And again, it will be this Friday at lunch. It's been approved for Flames Domain 4 credit as well as Kappa Psi and Phi Delta Chi credit.
So professional committee applications are going to be due Sunday, January 31st at 11:59. So if you haven't done so already, please look at the email for more details. In addition, we have our first outreach event of the year. We will be presenting about the career of pharmacy to the students of Pui Talk. It is Flames Domain 3 approved as well as PLS credit. So if you are interested, please look at the email for the sign up link.
Yep, thank you for that, Eunice. Very exciting. If you guys want to get more involved, especially if you're interested in the professional chair position, then definitely encourage to apply to the committee and sign up for that event. I believe we have more events coming out this semester, so really excited about that. But our next GBM will be hosted by our very own P1 liaison. It'll be on Monday, February 22nd. So that is the next time that we will see you all for our GBM. Yes, that is Justin. So definitely save the date for that one because he'll be talking about a very interesting topic. And our next EBM, for those who are interested in running for eBoard, you do have to attend at least one EBM. And our next one will be on Monday, February 8th at lunch. Okay, and then we did want to briefly talk about the requirements to run for the spring election for those who weren't able to make it to a membership drive 2 weeks ago. So to run for a position, you do need to be a paid member on or before March 6th. That's the 30-day deadline prior to the election on April 5th. To run, you do need to attend one EBM, as I mentioned just now. We will be meeting on Zoom from now on, and the links will be sent out in the weekly email prior to the EBM. So if the EBM is on Monday, it'll be sent out on that Sunday email. The bolded dates are listed there. Those are the dates that are still available that are prior to our election. The January 15th one has already passed. That one's been strike through. And then the April 19th EBM is after our election, so that one would not make you eligible. But there are 4 more opportunities to attend EBMs and get that requirement in. And you are welcome to come to more than one if you wish. And then the other requirement is to attend at least 3 APA events. These include GBMs, workshops, socials. So if you're coming to today's GBM, that would satisfy one of those events. And this includes events from both last semester and this semester. So it should be a pretty easy requirement to hit for many of you. For president and vice president candidates, we are instituting a requirement that you must have at least 1 year of service on eBoard, and you must have attended at least 5 APA events. And for professional chair candidates, you must have attended at least 1 professional event from either the 2 from last semester or some of the ones from this semester, or you can also participate in the professional committee, and that would satisfy that requirement. So if you have any questions about any of these requirements, please feel free to reach out to myself or any of the officers. On top of these, you will be expected to submit a platform for your chosen position. More details about that will be sent out in early March, which is when those will be released. With that, we will go ahead and invite our guest speaker to speak. Chi will help with the introduction.
Yeah, so for today, we'll be inviting Dr. Miguel. He has a PhD in PharmD degree from the Faculty of Pharmacy of the University of Lisbon. And he is also working as a current postdoctoral researcher at the Alzheimer's disease laboratory of Dr. Gary Landers at Indiana University.
All right. Hello, everybody. My name is Miguel. And as she said, I'm a PharmD, but I work as a postdoctoral researcher in IU School of Medicine. And first of all, I just want to thank Shi for the invitation to talk to this forum, which I think is mainly or mostly students, pharmacy students. And for that reason, I thought it would be interesting to share my experience through my PharmD, PhD, and then now my postdoc, since many of you are probably considering what to do when you finish your degree, what career choice to go, and hopefully this will maybe help you to think about how to go about this particular career, which is research in academia, but could also spark the interest in some of you, to do something similar. If you have any questions, or you need help with something that I can help with, you have my email, shoot me an email and we can talk. This will be a quick presentation. It's mainly just, let's say, an informal discussion about what you guys think, if you have any questions about any of this. So I'll just start. In 2004, I joined I enrolled in the Faculty of Pharmacy of Lisbon, the University of Lisbon. I'm Portuguese, so this is my home. This is a course that is like 6 years, and I think probably similar to the United States. We start with some general subjects, and then we move on to very specific subjects like pharmacokinetics, pharmacology, pharmaceutical chemistry, and so on. The thing with our course is that at the end, we need to do, which I think probably is also similar to what happens here, we need to do two types of internships. We need to work in a local pharmacy. I guess it's the equivalent to a CVS pharmacy here, which we learn a lot about public interaction with patients, how to manage stocks, drug distribution, how to help people, how to do their regimen, the drug regimen, etc. And I also had to work in a hospital, in a hospital pharmacy, and this was more focused on the pharmacokinetics aspect of that, the pharmacology, validation of therapies, all of that. We actually did some research there, not molecular biology research, but you can do a lot of epidemiology research. We actually did some research on that, and we presented this in a meeting. I mean, you have a lot of information when you work in a hospital. You can correlate, treatment outcomes with certain populations and all of that, which can be very useful for doctors, nurses, health professionals. By the end, by the very end of the PharmD, they also give us an option to do some research internship, and I took that opportunity, and our university actually gave us the opportunity to go abroad. And I'm not sure how is in your case, but I would recommend, if you have the chance to go abroad, to do, just take the opportunity to go outside of the United States to another country. I think it's super important. It will give you new perspectives, new ways to tackle a problem. You'll become more resourceful, more creative. It will expand your network. You'll probably easily adapt to when you actually start a job, because there's teams. Today's world is very global, so you have multidisciplinary teams from all around the world, and you can just easily adapt if you have all this background already. So I think this is extremely important. Go abroad, travel, try to get internships outside of your comfort zone. Anywho, as you may imagine, this was very rewarding to me. I really liked this internship. It was in a molecular biology lab. I worked in the Pharmacy Institute, and actually I worked in a project that years later, I didn't even imagine, but years later, I got my name in the paper from that institute. So it was actually very rewarding, this experience. So at the end of my PharmD, this was at the very end of my PharmD degree, I came back to Lisbon, to Portugal, and I knew I wanted to do something in research, in molecular biology, but I was not really very autonomous in the lab yet. I was a bit lost, because my course, maybe like here, was very focused on chemistry labs and pharmaceutical technology labs. That was a bit missing on the biochemistry, molecular biology side of that. So before I did something like a PhD, which maybe some of you also thinking about, I decided to that I need to learn more. I need to get more experience. So I took an opportunity in an institute in the university, which is called Research Institute for Medicines, to work as a researcher, just the researcher. And I worked there for like a year and a half to 2 years, I think. And actually, it was super good. I was super autonomous in the lab, and luckily, I got many publications with my name on it because I participated in many projects. And I was also lucky because I worked with PhD students that were already at the end of the PhD, which meant that, you know, they will publish a lot of papers, or like some papers, hopefully, you know, and I was lucky to work on those projects. So, you know, rapidly I could see the results of that, of, you know, doing this job. And when I felt that I had a good enough of a CV and actually experience, I then decided to apply for a fellowship for a PhD. So it was not immediately after my PharmD. And I really think this is the best way to do the PhD. When you get some experience, I think you need some experience after your course to do this. But so I applied for a fellowship, a PhD fellowship, which I got. And I started working in the same institute that I was working as a researcher. And this is the group that I was in. So this is me on the left here. This was my supervisor and the PI, the leader of the group. And like I told you, I seized the opportunity also to go abroad and work in Stockholm, in Karolinska Institute in Sweden, which is, you know, one of the most renowned institutes in Europe and in the world. Is very well known in terms of biomedical research. I worked here in this group, molecular and cellular exercise physiology, which was super good. Although I didn't do anything related to this, they had equipments that I needed, and it was very, you know, useful. They were very nice. I did not do also much of exercise there. But anyways, this is me here in the center, and this is the PI. Very nice group of people. Again, was very enriching, this experience, and I, you know, recommend go abroad, you know, try different things, different countries, etc. So in the end of my PhD, it was about 5 years. So it was from 2012 to 2016, more or less. I got 2 first author papers, which I think was good, and many other reviews. And just to give you an overview of what I did in my PhD, but I don't want to go into super hardcore science here. I just want to give you like very small overview of what I did was to work on brain cholesterol homeostasis. And in this diagram, you can see that cholesterol does not cross the blood-brain barrier into the brain. It cannot. So neurons rely on cholesterol that is synthesized by astrocytes, and then it shuttles through an ApoE lipoprotein to neurons, okay? So what happens to cholesterol in neurons is that excessive cholesterol is metabolized by this cytochrome P450, which probably you guys all know, because it metabolizes a lot of drugs in the liver, but this one is in the brain. It metabolizes to 24-S-hydroxycholesterol, and this compound here can be then eliminated, or it can travel back to astrocytes where it induces cholesterol efflux genes, you know, and kind of perpetuating the shuttle and the cycle of cholesterol towards neurons and towards astrocytes and like that. So our focus was here on this enzyme, the CYP46A1, because it had been related to Alzheimer's and other neurodegenerative diseases. And my project tried to answer how did the levels of this enzyme relate to neuronal like morphology and function. So the first thing that we notice is that when we change the levels of this enzyme, we change the activity of a particular group of proteins, which are called the small GTPases. And I know this seems like a complicated diagram, but you only need to retain a simple thing on this diagram, is that small GTPases, which here is raw, depicted as raw or RAC, while in the membrane, they're active. When they are not active, they translocate to the cytosol, and then translocate again to the membrane to become active. So this is like the cycle of activity of these proteins. So what we saw was that in animals, in the brain of animals that had more CYP46A1 here. I'm not sure how familiar you are with Western blots, but the quantifications are here. So anyways, so what we saw was that in the membrane fraction, there's more RAC1 in these animals, in the brains of these animals, and less in the cytosolic fraction, right? Meaning that when you have a lot of CYP46A1, there's a higher translocation of these proteins into active form. And these proteins, they interact a lot with cytoskeleton and a lot of things. So we thought, so what is actually happening to the neuron when we activate, when you have more CYP46A1, this enzyme, and you activate these proteins? So that for that, we explored how did the neurons look like? And you can see here, in red is a neuron that has a lot of this enzyme. So you can see that it has much more outgrowth of the dendrites, right? Secondary and primary dendrites compared to the control. You can see quantified here. And also you can see that in the dendrites themselves, you have this more of these little protrusions, which are called synaptic spines. Basically, it's where the synapses actually happen. Like these are the postsynaptic structures of that. So it seems like when you have more enzyme, you have more of these structures, meaning that you probably have more activity and more dendritic outgrowth. So these were basically the two main conclusions. I know it seems pretty simple, but it took me like 4 years to understand this. But these are the main conclusions of my PhD, summarized to 2 slides, which is, you know, yeah, it is what it is. And what we actually found pretty cool was that another group that was working on this years later found out that if you activate this enzyme, so this was like last year, no, 2 years ago, sorry, yeah. So if you activate this enzyme with an antiretroviral efavirenz, you actually have beneficial effects in a mouse model of AD, which is called the 5XFAD. And this for us was pretty cool because it means that we're actually working on something that is druggable, and you know, this is a preclinical evidence of repurposing a drug that could actually function in AD. So we were actually pretty happy with this, excited with this story. So throughout my PhD, I was very interested in this potential of, I did a little bit of basic biology, but I was very interested in the potential of translating that into something to the clinic. So this was something that I looked for in my postdoc. So it really depends on what you really are, motivated about. I really like this molecular translational part of science. So that's what I tried to look after when I looked for postdocs. So I looked for postdocs abroad. Again, I really like to travel. Someday I will settle in a place, but I really like to travel, and I think you can gain a lot of with that. So I looked for postdocs in the United States. And I happened to communicate with the Dr. Gary Landreth, which is like a very renowned scientist in the Alzheimer's field. And I really liked their lab because they had published some papers. And again, I think you should, if you are interested in joining some lab or some institution or something like that, you really need to try to figure out what I've done before, so you can actually see if they complement with your interests. So I knew that they had published some papers about repurposing drugs in AD mouse models, and that's exactly what I want to do and discover new therapeutic targets. So I sent them an email. It was pretty quick. I did a Skype interview, sent my CV, and he liked me and he hired me as a postdoc. So I went to Cleveland, Ohio, in Case Western Reserve University. And then we eventually moved the lab to Indianapolis, where we are now, in the Start Neuroscience Research Institute here, Indiana University School of Medicine. And one of my focus is, as I said, is to find new therapeutic strategy for Alzheimer's disease based on immune modulation, namely microglia, which are the brain resident immune cells. I'm pretty sure you already know that. Anywho, So I'm focused on trying to find novel targets for AD, but also at the same time, I like a lot drug repurposing. I think that's a very promising field. And I think you're aware of this concept, right? You know, in pharmacy. So so one of the things that I want to explore is how can we use already FDA approved drugs to you know, target some of these novel pharmacological targets, and because the translational process will be much faster, right? So it gives you a timeline that is smaller for the drug to be in the market for a particular disease. So this is something that I really like, and you know, I am focused on. Another thing that is important, and it's grants. And grants is something that if you go to academia, it's something that will, you know, it's basically how you survive. And if you're really interested in going through this career choice, academia, and even I think industry, I think they probably need to write grants. It's something, it's a skill that you need to, to learn, and if possible, even in your PhD, you can start writing some grants through your postdoc. You definitely have to write some grants, and that's a very important skill to have if you really want to continue into this career choice. I mean, grants are, you know, the way that you survive. It's exciting actually to write grants. It's a bit frustrating because you don't get all of them. Most of them you don't get. You get some, but you know, it's a part of the process. Another thing that I forgot was also that throughout the PhD and postdoc, you will supervise undergrad students, which is, again, it's a skill that you will need to learn how to do if you really want to continue with this career choice of academia and research. I mean, even probably other career choices. You will need to learn how to, you know, teach, supervise, you know, transmit, you know, knowledge, so that, you know, the people under you will learn and do their job correctly. Okay, so now back again a little bit to science, not so hardcore science, but let's go. So why are we focused on, you know, microglia? One of the microglia has emerged as one of the major players in Alzheimer's disease. And one of the reasons for this is because many risk factors for Alzheimer's that have been, you know, discovered now are related to immune function, or are microglial genes. And you can see them in blue here. One of the most known is TREM2. This is a super hot topic in the field. You have other here, CR1, CD33, but there's many others that have been, even the INP5D here should probably be blue, but there's many others that, you know, have been recently also found that are not in this graphic here. So this kind of like led the field to think about microglia as a major player in AD pathogenesis and even in disease progression. And so what does microglia do in the brain of an Alzheimer's person with Alzheimer's disease? So it seems that one of the major functions of microglia is that, so you know that in Alzheimer's disease, there's these plaques that are A-beta plaques, extracellular, right? And so what happens with microglia is that you can see here in light blue, it's an A-beta plaque, and in white is microglia. So you can see that microglia basically does a barrier around the plaque, and it stops the spreading of toxic species that come from the plaque and, you know, then are toxic for neurons. So it provides that little barrier there. You can see that a little bit better here in another mouse model of AD. Here you can see that in red, there are plaques, and in green, they're microglia, and the yellow is basically the two of them, like the microglia doing this little barrier, right? And these are two mouse models of AD that create plaques. These are mice. So, and just to highlight the importance of these genes that I just talked about, and I'm just going to show TREM2, but you know, many of these genes are important. Deletion of TREM2, which is a microglial receptor. So it's just one microglial receptor that you delete. You completely change how the pathology presents itself. And you no longer have this tight barrier around the plaques, as you can see here. So you mostly see red, 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 a little bit green, which means that microglia is not doing what they should normally, and they are releasing more toxic. The plaques are releasing more toxic or neurotoxic species that leads to neuronal dysfunction and neuronal loss. This is just to highlight why we're focused on microglia. And you see, just by taking out a small receptor microglia, you have a huge impact on how the pathology presents and progresses. So what we want to do with microglia is try to stimulate microglia in a way that is optimal for Alzheimer's. So you don't want to stimulate too much, right? You don't want to activate microglia too much, because that would be detrimental for the brain also. But you probably want to still want them to perform their function, like the protective function. So there's a sweet spot here. There's a window that we need to hit. And it's in that window that we are looking for targets and receptors or targets that we can target with some drugs. In my case, I want to target this with FDA-approved drugs, but this to get microglia to do something in this optimal activation window that is good for Alzheimer's, or that will give benefit effects to an Alzheimer's patient. And I'm almost over. So, but it has a last slide. What I want to say is that throughout my experience, and I think I'm pretty quick actually in the time, but throughout my experience in the labs that I've been, one of the things that I noticed, there are mostly multidisciplinary research teams, but there are not a lot of pharmacists in these labs. Of course, there's pharmacists in R&D, but many labs don't have a lot of pharmacists. And I noticed that as a pharmacist, we have a unique perspective in biomedical research, which I think, at least I didn't think about this a lot, I just realized this after years being in labs, is that our way of thinking is very different from other people with other backgrounds, but they complement each other. And for instance, we always have concepts like PK/PD in our minds. What is translation, what is not? Is this a druggable target or not? So this is like in depth in our way to see things. At least for me, it's like I was hammered with this so much for 6 years that it's just like the way I think. And this is actually an advantage. And this is something that you should highlight and emphasize when you apply for things, because not many people have this perspective that I think we do, and complements really well. I mean, of course, depends on the objective of the lab or the industry or whatever thing that you're applying for. But I think this is something that at least I forgot in the beginning when I was, in the PhD, when I was starting. I didn't even think about this. So, but this is something very important and makes a lot of difference when you are designing experiments and the outcomes that you want to measure and stuff like that. So, this is something that you would emphasize when you want to apply for jobs. Now, if you want to apply for a job that, obviously that is already a lot of pharmacists in, like a pharmaceutical industry or something, then within the background of pharmacy, you need to differentiate yourself with other things. But if it's a multidisciplinary lab, I mean, you can really emphasize this, I think, in my view, this thing about being a pharmacist and how pharmacy really makes you unique in terms of the biomedical research. And so that's it. And and if you have any questions for me, if you want to discuss, if you have questions about what career, about this career choice, money, maybe it's important for some people. Shoot me an email, or you can just ask right now.
We had a question in chat. You mentioned your interest in translation from benchtop science to clinical setting. Do you yourself get to do any clinical types of work, or is mainly lab work that could be applied to the clinical setting in the future?
Yes, that's a good question. Yes, so I myself right now, I don't do any clinical work. So what I do is preclinical. So I basically do mice work. I could do some PK/PD in mice and all that. But myself, I don't do what we are planning to do. What we can do is, when we have a robust body of results of the preclinical, so with the animal results, we try to get funding in doctors and pharmacists or whatever to apply for a clinical trial, or a pilot clinical trial, which I probably won't do it myself. I would be just involved in the back office, kind of, not directly, but maybe. I mean, if you're a PharmD, maybe you could, if you wanted, right? Maybe you could apply with a clinical trial doctor, and probably if you like that, it's possible, I guess, for sure.
Follow-up question that's probably easier to say out loud than to type it out. All right. So with the dual degree, there's a lot of sort of time invested, obviously. Do you ever, just from all the time you spent as a postdoc now, are you finding that you're able to really get a use out of both the expertise you got from your PharmD and also the experience you're getting from your PhD, or do you feel that your work, you might be pulling more from what you got from doing your PhD over your pharmacy degree, or vice versa, if that makes sense, because you're investing time for both?
Well, to be honest, I think I'm taking more steps from my PhD into my postdoc. But again, I think pharmacy is always there, and of course you invest time in both. But pharmacy is always there in terms of what I was talking about in the end, about how you view, like it's like your DNA, right? I mean, I see it has my DNA. I cannot really separate myself from everything that I learned in the pharmacy school. And although maybe I cannot see it directly, because I learned how to do Western blot in my PhD. So I do the Western blot in my postdoc, right? So very direct things in the lab, yes, I take a lot of my PhD. But in the concepts, no. In the concepts, I would say 50-50. In the concepts and how I view science, and how I view how we can make better science, and how can we translate our findings into clinic, I get a lot of what I learned in pharmacy, to be honest. It's not just because we are in the pharmacy meeting. It's really true. It's really true. And it took me a while to realize this. It took me, even after, it was like at the end of my PhD that I actually realized that. And I was taking a lot from my pharmacist background, really a lot. And even the way you talk to doctors, imagine that you're doing a very basic research and you have to communicate to a doctor because you need, tissues, or you need to collaborate with some patients. A pharmacist will, I think, have a much better understanding and communicate much better to a health professional than, let's say, a person that has been always in the lab even throughout the course, right? So yeah, I think, I would say like 50-50, although you might not notice it at first, but I would say it's always there. It's a part of who you are as a researcher, in my case, in DNA, for sure.
There's a question in the chat. Were there any major difference you noticed from working in and out of the States?
Yes. Right. This is a good question. Yeah, I don't want to get into much details here, but there is a lot of differences. In the lab, not that much. Let's say this. In the lab, working in the lab, there's not much differences, but always, culturally, it's a bit different. Now, for instance, in the States, I think you have better conditions in terms of salary and in terms of medical healthcare, because it's all covered. And in many other countries, researchers and postdocs don't really have this status. So you receive less money, and you're not as protected as in the United States. It might seem surprising, but this is true. Regarding the work itself, I think things work, if you want, things work faster here in the United States. If you have a fast pace, and things will work at that pace. In Europe, for instance, it was not bad, but I think, the pace was not that fast. There were other advantages, but, as I said in the presentation, you really need to experience, I think everybody should experience at least once in their life to what it is to get out of your comfort zone and go out to another place and work there, and you'll get what I'm saying about the differences in places. It's very, very rewarding, I think. But yeah, totally. United States is a very good for science. work as a scientist, United States, I mean, it's probably one of the top places to work at. Yeah, for sure.
There's one additional question. Any advice you may have for those people, for those of us looking to go out?
Yeah. Enjoy. No, I just joke. So, I mean, really depending on where you want to go and what you want to do. So if your interest is to do, I don't know, an internship in a pharmacy somewhere or part of your PhD somewhere, my advice is to stay as long as you can, because you probably will come back. You know, choose somewhere that you really feel that you will give you a different experience from the from probably where we are now, the United States, and just enjoy. This seems like super like a cliche, but really just go meet the other researchers or professionals. Go out, enjoy the network, just network a lot. Just take enjoy every single thing, because it will be over one, and then you'll probably gonna regret if you didn't really took advantage of that.